Okay, so we're proud of some another more technical difficulties. We've been talking about Taq polymerase. And what Taq polymerase is, it is a DNA polymerase that comes from the Thermus aquaticus, which is this bacteria that lives in the hot springs of Yellowstone National Park, very, very, very high temperatures. And what is true of bacteria that live in high temperatures is that their polymerases, their enzymes, are going to have an optimal temperatures that are also going to be very, very high. So once we cool it down to 55 degrees Celsius, we can then add Taq polymerase to help with these primers, and it's going to help build new strands of DNA. And what type of DNA is it going to build? It's going to be radioactive probes, because what are they made out of? We added radioactive nucleotides so that we can trace it. And I also want to remember the 5' to 3' rule. But when we're making these probes, so we're making copies of these probes. What we have here is we can heat it up, separate them out, denature them, and then cool it, allowing the primers and Taq polymerase to come in. And we can literally, through this process, make thousands of copies of our probe. Thousands of radioactive probes can be made. So what do we do with these thousands of copies of radioactive probes? Well, we can put them back into the ASI wells. We can apply a photographic film to it. We put the photographic film on there, and the radioactive probes are going to cause a photographic film to develop. I mean, think about an X-ray. What are you having there? You have this sheet of film, you stand in front of it, they shine X-rays through your body, and it shows you what's where, because radioactivity allows photographic film to develop. So this right here that we see has been marked on by the radioactive probes. That's going to point to us to where in the ASI wells the gene of interest is, the bacteria with the gene of interest are. And I want to make a point about when we're talking about polymerase chain reaction. One of the things that's useful about this is, and he just gave us a large overview, but polymerase chain reaction can be used to make copies of specific DNA fragments to be inserted in vectors. This way we can make, think about it, if we're making copies of just a specific probe, we can make copies of specific DNA fragments that we can insert into vectors. That way we don't have to cut up the entire genome of something, and we don't have to do this whole multiple screening process. Well, I don't know what's what. This way we can have an assload of specific pieces of DNA fragments that contain the gene of interest. And this saves a huge amount of money and time for people who are working in synthetic biology or genetic engineering. So we apply it to the film to develop, and so that's, as a whole, that's how we go from having a gene from a prokaryotic cell, and we insert it into another prokaryotic cell. Now, I mean, that's cool and all, but that's not really going to provide us with a whole lot of options in terms of clinical application to that, because I mean, what good does it do for us if we have something that prokaryotic cells have made? So what's much more, I guess you could say, clinically applicable is to take a eukaryotic gene from a eukaryote and insert it into a prokaryote, so that we can, because prokaryotes have a very short generation time. They populate like Mexican Catholics. They have 24 hours. We can have billions of them made. So we can make mass productions of things that, people make. And so the problem, though, being that DNA on prokaryotes is non-intronic. There are no introns on it. It's just one circular molecule, and it goes round and round and round. It's all coding for protein. It's all exonic. Whereas humans, 95% of our DNA is intronic. 95% of it. So if we were to take a eukaryotic gene and we were going to insert it into a prokaryote, what we would get would be something that would be, because of these introns, it would be much larger, it would be deformed and dysfunctional. And this makes me think of, and this is a horrible drawing, of Bizarro from Superman, who, Bizarro was kind of like a copy of Superman, but he is all deformed and mutated and things like that. And that's what would happen if we took that, because the prokaryotes can't distinguish what's an intron and what's an exon, and they'll just read it as so. So what do we have to do? Well, what we have to do is we have to harvest, I like that word, mature messenger RNA from cells that make the polypeptide of interest. And once we do that, so we take the messenger RNA from this, and that makes this polypeptide of interest. Now we have mRNA. Because mRNA, mature messenger RNA, what's something about it? It has no introns. It's 100% exon. All that's on there is going to code for a protein. The problem being, though, that this is an RNA. RNA will not fit in the plasmid because the plasmid is made out of DNA. So how do we do this? How do we get this messenger RNA to formulate DNA? Well, we got to expose it to reverse transcriptase. Reverse transcriptase is something that viruses use to undergo RNA-directed DNA synthesis. And this is how viruses have literally hacked their way into the human genome, is because of they contain reverse transcriptase. It is a DNA polymerase. It makes DNA. So what we end up with is a hybrid molecule, which is half DNA and half RNA. So here's your, I don't know how clear the color is, but this blue thing here, this is messenger RNA, and this green thing, this is reverse transcriptase. So we start off with just a blue messenger RNA molecule, and then as we start, reverse transcriptase moves along, it starts to transcribe, it starts to polymerize the formation of a new DNA strand that is complementary with this messenger RNA strand. After that, what it's going to do is it's going to go back up through here, and it's going to, I draw these are supposed to be scythes cuts up, it degrades the messenger RNA. It degrades the messenger RNA, but at the same time, as it's doing that, it's going to lay down DNA, it's going to lay down new DNA template strand that is complementary with this DNA strand. So what your end result